Right. Perfect. Thank you very much, Dr. Byrne. And thank you for the Tools for Polyploids crew for setting me up right after the GWAS Poly talk. It was a very convenient time to give this presentation because now I'm going to be giving this example, this applied example of its use in Tetraploid Blackberry. So yes, hello from Arkansas. We'll be talking a lot about GWAS Poly, a little bit about some other tools that we used as well in performing the first GWAS and Tetraploid Blackberry. So let's jump in. So yes, my name is Mason. I'm a PhD student at the University of Arkansas with the Blackberry breeding program, which began in 1964 under the leadership of Dr. James Moore. And then from then, it was passed on to Dr. Clark, who had tremendous success with the program, very full career, and has made a lot of improvements to the crop, and is now handing it off to the third generation of breeder, my advisor, Dr. Worthington. So we're on the third generation, and now she's ushering in a new age of molecular tools, and it's been a privilege to be on this project and to participate in that as we perform this project. So historically, some of our breeding goals are shown here. Take a quick look at those. We'll talk about them a little bit more to come. I'm going to open the door. My dogs are making some noise. Apologies for that. Anyway, the question that we weren't able to ask early on that we are now, especially with a lot of these emerging tools and technologies, is what are the genetic origins for our genetic gains that we've made in some of these breeding goals? So how can we speed that up? How can we use markers to assist us in our pipelines and perhaps even genomic selection further on down the line? So today we'll be talking about the GWAS. So how are we going to do that? Well, our breeding program lives in the Fruit Research Station in Clarksville, Arkansas. We get cold, icy winters. We get hot, humid summers. So a lot of seasonal variation affecting our plants, and that's our environment. We've collected fruit. We're going to push all that, and a lot of you who work with applied breeding programs and do this, that this phenotyping aspect of things is where a lot of the work is. And so we spend a lot of time working on imaging pipelines, measuring fruit morphology, seed morphology, fruit composition, and really just getting all this wonderful phenotypic data on these quantitative traits. And we'll be plugging that in with some population structure analyses and into GWAS Poly, looking at linkage disequilibrium and some of those new updates with GWAS Poly. And we'll go ahead and speed through this. So those who have conducted genome-wide associations know that you don't simply do a genome-wide association study. There's things you need. You need a reference genome, and we've got a nice, pretty new reference genome, the Rubus argutus one, that is it's a diploid reference genome, but it represents a important founder for a lot of our germplasm, particularly our primocane fruiting materials. And so it's very relevant to us, very nicely annotated and chromosome scale. So that's very nice. We also need the work of a lot of students and some high throughput phenotyping methods where we can implement those. And we've got a few examples of those here. We're measuring things like texture, berry composition down there on the right. We've got myself and two other master's students on this project, so we had a lot of hands, and my advisor was able to secure a nice AFRI grant to be able to fund the work on this project. And we're trying to implement some new image-based phenotyping methods as well, pull basically as much image data as we can from a single picture of fruit. So all that's going to get plugged into our GWAS model. And you can see, just sort of scan this list of traits that are involved in 3 different years in a single location, and 2 replications within the harvest season, separated by a week. So we try to replicate things temporally within the year and across years, and we'll have about 301 genotypes with a lot of imbalance in there between years. But basically, fruit from every advanced or any breeding selection that we can get our hands on in our program. So next we need adequate read depth to be able to handle tetraploids. We used the same service that I think Gibran used, Rapid Genomics. We used the Capture-Seq platform. We got 35,000 Capture-Seq probes, which yielded 125,000 SNPs, filtering for quality, trimmed down to about 80,000 SNPs. And those 80,000 SNPs had about 150x read depth, so pretty good statistical power for estimating our dosages. And our SNP calling was performed in UpDog, which we had very positive experience with, very easy to scale it up to a high performance computing system, and then to take that data and plug it into GWAS Poly. So we've got all the things we need. This is a familiar plot to everyone based on the update that we just got with GWAS Poly. The linkage disequilibrium, what we were seeing was just a little bit shorter than the one that Dr. Endelman just showed. So it may extend as far as 7 megabases for some loci, but we know from that update that maybe we should trim it back a little bit, maybe like 4 megabases, something like that. So it's possible for our QTL to be relatively distant from our causal gene. That's sort of that double-edged sword where it could be far away. You've got these wide peaks. You don't know exactly how close you are to a gene necessarily, but it allows you to search a little bit further. So now, before starting our full association analysis, we did conduct heritability estimates as well, our broad sense entry mean based heritabilities conducted over a year. So we experienced relatively high heritability for seed morphological traits, relatively low heritability for a couple, including berry width, not highly heritable, and soluble solids content, which is basically sugars. So a lot of environmental influence there, but overall pretty promising heritability estimates for conducting a GWAS. Finally, we have all of our phenotypic data. We have our SNPs we talked about, the filtering, all of that is cleaned up, ready to go. We've estimated BLUPs, and now we're going to combine it into this GWAS Poly model. And actually, Samuel had asked in the chat previously about the Q plus K model. And so we're about to arrive at a comparison there. We did include a Q model using a K equals 6, so 6 clusters from structure, which our statistics had indicated was ideal. So those are the fixed effects based on clustering that we're going to include in that top model. We've got our Q matrix and the bottom model without. And so we're solving for our marker effects. And so let's jump in. First, we'll talk about thorns. We're going to visit this trait. It's been a very historically important trait. I mean, when we started seeing thornless blackberry, that really changed a lot about the industry, and now we have a panel that's about 90% thornless. And regardless of how many thornless we have, we can see that genetic gain occurring in the thorns phasing out, but we still found a very high effect QTL. And it was very close to one that had been previously identified by Tagi and Weber in 2020. Which was at 34.7 megabases, about 1 megabase away from where we identified a peak. So that's very promising to see. They had stipulated that it was probably a MIB16-like transcription factor. So a little bit of a sanity check there, and to show that this is working. And we see both the K and the Q plus K model being pretty much the same. And it'll be pretty much the same for a lot of traits. So another one that's important to us now, perhaps more for recent developments, is primocane height. So for those that are unaware of what a primocane is, just real quick, blackberries they are perennial plants. They have a perennial rootstock, but they actually have biennial canes. And wild blackberries typically only fruit in the second year. So those are called floricanes, two-year-old canes. The first-year canes, the primicanes, are capable of fruiting now in certain genotypes, and that's a trait that's been emerging since the early 2000s. So we've got primocanes, and we're starting to see dwarf varieties that are being developed by our program that maybe have a niche in sort of houseplant, small gardening kind of situation, or in indoor commercial growth settings. So this trait is very relevant to both of those. We identified something very promising on chromosome 4. I mean, you see those two peaks, chromosome 4 and 6, but chromosome 4 occurred fairly close to the gibberellin 2 oxidase gene, which is a green revolution gene. This is a very well-known gene for these sort of dwarf situations. When inserted into the Arabidopsis genome, for instance, it leads to dwarf plants. It's been very influential historically in a lot of crops. So we're excited about that. We hope to follow up and to see if perhaps that's our winner with our brachytic dwarfism. And chromosome 6, we still need to dig around a bit. So fruit pH, also very important to us. For a lot of people, blackberry is defined by its very tart flavor. That's one thing we're trying to balance a little bit better in the breeding program. So knowing what the underlying QTL for that is important to us. So it has a strong negative correlation with titratable acidity, just another metric essentially for measuring something, I won't say the same, but it's similar and highly correlated. And that's reflected in our Manhattan plots. On chromosome 5, this one's for pH. We found one at 4.4 megabases. There's a few possible candidates, either these malate synthase sort of genes or ALMTs, which are transporters of acids, moving acids into the vacuoles. So perhaps something to do with synthesizing or moving acid around. And so we'll have to follow up with that to validate those. Now, fruit firmness is one that's close to my heart. I did a lot of the work on collecting this data. Now, unfortunately, we have a bit of a messy peak, and this is a situation we run into where that K and Q plus K model actually did make a difference for us. So the K model, we observed more significant results. Q plus K just barely below that significance level on that peak that we saw towards the beginning of chromosome 2, around 6.3 megabases. And there are promising candidates close to there, particularly a homologue of polygalacturonase. So if you've ever done any reading on fruit texture and post-harvest textural changes, polygalacturonase is sort of the number one, perhaps the most important enzyme with this. What it does is it breaks down the pectins and can soften those cell walls, leading to softening of fruit. For instance, the difference between a melting flesh and non-melting flesh in peaches.
3 more minutes.
Okay, almost done. So we're going to try and confirm this with a RNA expression sort of study. I'm going to skip on a little bit. Fruit length, we don't have anything super exciting to say about specific candidate genes, other than on this chromosome 6, towards the beginning of chromosome 6, we expect that this has something perhaps more to do with flowering rather than our traditional candidate genes for fruit size in maybe like a pome or a true berry sort of fruit, because those individual drupelets are ovules, and the number of ovules sort of translates to fruit size for us. So we've got work to do. Perhaps some of you have suggestions on where to look. 100 seed weight and width to length ratio. 100 seed weight means larger seeds, so we've got a correlation there. A couple QTL have really one particular for each of these two traits. And the only thing I really have to say here is that there are some homologs that are related to size and shape in rice, which has been a very important trait for them, because larger rice, higher yield, so perhaps there's something similar to be said there for our crop as well. So overall, we found highly predictive QTL in most traits that we sought out to look for. This is really an ideal situation. Very happy with the success of this project, other than in soluble solids and berry width. We mentioned those aren't highly heritable. And the most exciting story is yet to be told. I'm afraid I've been holding out on you. The primocane fruiting QTL is very exciting for us. We have a LOD of 60, but we're keeping that a little bit under wraps until we get that publication out. So we want to develop and validate diagnostic markers for high effect QTL, explore these candidate genes a little further, and we want to train a genomic selection model using this data, which is going to represent a perfect training data set for that to come. So looking forward to learning from you all on some of those genomic selection tools that are emerging. So thank you, my advisor, Dr. Clark, for constant advising, all the farm crew without which this couldn't happen, my fellow students, Carmen Johns, who's in the call right now, and everyone else, lab manager Lacey Nelson, I believe, is also in here. Thank you all, and thank you, Tools for Polyploids folks, for being so helpful and organizing this and doing what you do. I appreciate you for listening. I don't think I have time for questions, but maybe in the chat.